and thanks very much for the opportunity to speak to you about some of the work that we've been doing. Just the talk outline is really, for many, many years, we've really been searching for correlates of protection to HIV and using maternal infant transmission as the model to study this. And really that's looking at natural resistance to HIV infection in the infants, but then also the ability to look at the HIV infected mothers and look at issues of attenuation of infection in the mom. So it allows us to look at both infection as well as susceptibility to infection. I'm going to give a little bit of introduction, tell you a little bit about innate and adaptive immunity, because these are all immune responses we're looking at, and also then host genetics. I'll explain a little bit about the model and why we think it's a good model to use to study protective immunity, and then some of what we've found. So why are correlates of protective immunity important? Well, one of the most important, ever important goals in HIV vaccine research is to understand what correlates of protection are. If you can understand what they are, we can then decide on rational approaches to get the right kind of responses that will protect individuals who get vaccinated against HIV. And a lot of the work that has been done has presumed that certain responses are protective when we don't know that they truly are. And that's often been the failure in us getting very successful vaccines. So it remains an important ongoing question. Talking about innate and acquired immunity can be present at birth. The textbooks will tell you it's not present at birth and only gets acquired once the child is born, but that is not true. There are a lot of exposures that happen to antigens, to allergens in utero while the fetus is in, while the baby is in the mom. There are exposures to various proteins. But when a baby is born, through various antigenic exposures, the immune response gets boosted by repeated exposures, and the cell types that are responsible for the adaptive immune response are your T cells and your B lymphocytes, and obviously specific antibodies. When we talk of innate immunity, this is, I'm going to say, apparently not dependent on memory of previous exposure, because now very recent data is suggesting that that is not true. So a lot of the textbook dogma is kind of being challenged at the moment in terms of innate immunity. The cell types that are involved here are dendritic cells, natural killer cells. I highlight natural killer cells because I'll be talking about them. Phagocytes, which are neutrophils, monocytes, and macrophages, and then various soluble factors, complement, acute phase proteins, cytokines, again I've highlighted because I'll be talking about chemokines, which are really cytokines that actually have chemotactic ability, and then antimicrobial proteins. And the interesting thing is that people have generally compartmentalized, and I think that's often the failure in terms of trying to understand immunity, is because these are dependent rather than independent immune systems. And a lot of what we're seeing in the innate immune response now is certainly suggesting that it's actually dangerous to compartmentalize to such an extent. So just to show you here, and these are very recent findings, basically in 2009, as a result of a number of mouse studies that were conducted, the traditional model of how we see innate and adaptive immunity developing is being challenged, and a revised model has been proposed. So what would normally happen with an immune response is when you first get exposed to an infection or to an antigen or something foreign, you have very quick responses by your innate immune response, your myeloid responses, your NK responses. So this is magnitude of your response here on this axis. But what happens with your T cell or your adaptive immune response, you have this expansion here of your primary T cell and your B cell responses. It then drops off, and this is a period of really where you have the development of B cell or T cell memory. And then once again, when you have secondary exposure or secondary infection, the same thing happens with the innate response. It's always been considered as you have an increase in activity, it does what it needs to do, and the response dies down. There's no memory of that. Whereas your T cell response then rises, and it's usually a higher magnitude than what happened at first. However, because of these recent findings in terms of mouse models, the innate immune response and natural killer cells in particular have been shown to have a rise and have, not to the extent of the magnitude of your T cell or your B cell responses, but also have the ability to develop memory and then have a higher magnitude upon re-exposure. So that certainly has challenged the thinking that there is the typical classical textbook thinking. And I talk about NK cells because right now there are a lot of very interesting things happening on that front, and I'll share some of our data with you on that as well. So just to explain a little bit about acquired immunity, because I'll be talking a little bit more about peptides and how cells interact, and just to have some basic understanding. So it's really your CD4 and your CD8 T cell responses. So what happens in the case of a CD4 T cell response, you have professional antigen presenting cells, which are either your monocyte macrophages, your B cells, or dendritic cells that see exogenous antigen. They see antigen outside of the cell, gets taken up by the cell, processed, and then placed on what are called class 2 human histocompatibility molecules, so HLA class 2 molecules. And the peptide gets placed in this little groove here and presented to the T cell via the T cell receptor. So it's basically your peptide sits there. So your foreign peptide then, this interaction between your antigen presenting cell and your CD4 T cell culminates in an expansion of these cells, so an increase in proliferation, the production of cytokines, and then because they're helper cells, they provide help for B cells as well as cytotoxic T cell responses. The CD8 T cell, or a cytotoxic T cell, here your antigen presenting cell is any infected cell. So any cell that becomes infected, for example, by a virus, you'll have your virus, you'll have bits of protein chopped up and then presented together with class 1. So these are HLA class 1 molecules on the surface of your CD8 cells. Your peptide placed in the groove there, and once again your T cell receptor on this cell interacting there. And this interaction results in the recognition of the cell as foreign or infected, and your cytotoxic T cell will then kill the particular infected cell. So I'll come back to some of the responses that we look at, and because it requires those peptides, I think that basic understanding is important. So the interesting thing that happens in the context of pregnancy and immune responses is that this is a state of unique dysregulation that happens between the innate as well as the adaptive immune system. And you have a specific adaptation of the maternal immune system for a very important reason. It's really to protect the fetus from T cell attack. So you find that there's this immune tolerance as well as immune suppression, because the T cells will recognize some of the antigens on the child as foreign. And those are really the component that is really inherited from the father. So there's some recognition of self, but there is also recognition of non-self, and that's what the child has inherited from the. So you want to reduce the ability of this particular part of the immune system, otherwise you'll get this attack, and this can lead to spontaneous abortions. So the other reason why there's an adaptation is that you have to provide strong antimicrobial immunity to then make up for the fact that the T cells are kind of immunosuppressed to some extent. And this is through immunomodulation and through the enhanced innate immunity. So there's this change in the immune response in a pregnant woman, basically to protect the fetus, but to still provide antimicrobial immunity. So you have downregulation of T cell mediated responses, and these have often got a Th2 versus a Th1 type of bias, and activation of your innate immune system. So during the process of pregnancy and very close to the time of delivery, you have a huge expansion of cells, such as monocytes and neutrophils, basically to provide this kind of antimicrobial activity. If we look at the immune system in early life, the neonatal period, that's the first 28 days of life, was marked by very, very high susceptibility to infections. And the first line of defense of the infant is really their own innate immune capacity. So the innate immune system is quite important to them. If you do a direct comparison between the immune response, even the innate capacity of an infant versus an adult, you'll still find that that's deficient by comparison. The other form of defense for the infant is really through specific antimicrobial antibodies, which are received through passive transfer from the mother across the IgG that crosses the placenta, or they may get IgA, which comes in the form of through breastfeeding. And this helps to protect the child to some extent while their T cells, or while their own immature adaptive immune response starts being sensitized to seeing different antigens over time. So essentially, obviously in HIV infection, we know that sometimes the antimicrobial antibodies, there's deficient passive transfer. So certainly in women that are more ill and that have low CD4 counts, they sometimes don't effectively transfer antibodies to their children, and that may also then make the children a little bit more susceptible. So in terms of the infant's own immune system, they're very reliant on their innate immune capacity early on in life. And it's very, very likely, and therefore seemed like a good to look at innate immunity in terms of prevention of perinatal HIV infection. So a little bit about the model that we've used. So our approach to trying to study protective immunity has looked at, we know that there are a large number of infants that even in the absence of any antiretroviral treatment to try and prevent perinatal infection, a large proportion of infants still remain uninfected. And the question is, why do some of the infants escape infection while others succumb to infection? And if we can get some of those answers, we can help to identify critical components of protective immunity. We also know that infants acquire infection either during pregnancy, during delivery, or through breastfeeding. So you have all these different modes and probably different susceptibilities because of the different scenarios in these children. So really it's a natural experiment, because if you look at it in this way, for example, in utero, there's definite evidence, there's a lot of evidence of immune stimulation that occurs in HIV-infected, or infants born to HIV-infected mothers. And this is both specific and non-specific activation. So you can look at it from the point of saying this is priming or vaccination of the infant in utero through virus or proteins. We often, at birth, if an infant's infected in utero, obviously you can't look going forward, but the infants that are uninfected, we can look at immune responses at this particular time and then ask the question, do they become infected during delivery or intrapartum, or possibly through breastfeeding if breastfed. In the past, we've worked on aspects of acquired immunity. We've looked at CD4 T cell responses to particular HIV peptides, and it has been shown in 2 studies, including one of our own, that infants who at birth are uninfected, but they have a response to the envelope region of HIV, none of them subsequently become infected, either intrapartum or through breastfeeding. Our study only addressed the question of intrapartum transmission, because a lot of the studies that we've done with the perinatal HIV research unit have been on mothers that elected not to breastfeed. So for example, here, this was the figures from the breastfeeding study that was done down in Durban. Infants who did not have these responses, 17% became infected, whereas those who had responses, none became infected. I'm going to be focusing this talk largely on innate immune responses and more recent work. So again, this provides a model where infants are naturally exposed to virus once they're born, either through intrapartum or breastfeeding, and it allows you to then ask that question. So why is mother-to-child transmission a useful model for the study of protective immunity? Well, we can evaluate both the mothers and the infants. We can look at viral as well as immunological factors. We can study transmission, but we can also study control of infection in the HIV-infected mothers. Because the mother and child have got genetic similarity, so some of the genes from the child has copies of genes from the mother as well as the father, we can actually look at host factors and genetic aspects, particularly in this model. HIV exposure can be defined. We can measure viral load in plasma, in breast milk, and vaginal secretions. Infant responses can also be defined at and close to the time of exposure. So you have more control over when you can measure responses in relation to exposure. And the timing of transmission can be estimated. And this is by using polymerase chain reaction, PCR, to determine that. So just to give you an idea of a scheme of the sample collection and assays, for example, what we would do in a standard a cohort study, we would often collect cord blood as well as mother's blood, and then infant blood at within 48 hours, 6 weeks, and 3 months. HIV DNA PCRs would be conducted at those various times. If an infant is positive at 48 hours, remains positive at the other time points, it would be an in utero infection. If negative at 48 hours, positive at 6 weeks, and ongoing, then the child would be infected intrapartum. And as I said, our studies have really focused on in utero and intrapartum transmission. And then various assays. You can do immune function assays, various phenotypic assays. We can look at host, we can look at virus genotyping. So I'm going to talk about 2 correlates of protection that we've done quite a lot of work on, and they're both part of the innate immune response. The first is elevated production of a chemokine, which is known as CCL3 or MIP1 alpha, and also a relationship with higher gene copy number of the gene that codes for that. And the other is unusual natural killer cell responses to HIV peptides. And these were responses that were discovered when we were actually looking at, our intention was to really look at adaptive immune responses. And this was a surprise finding, and I'll tell you about that. Okay, so firstly, the chemokines. These are the natural ligands for the chemokine receptor CCR5, which we know is a major HIV co-receptor for CCR5 tropic HIV isolates. And the natural ligands are CCL3, 4, and 5. So this is the newer nomenclature. So people may know it by MIP1 alpha, MIP1 beta, and RANTES. and they all bind to CCL5, and they can all block HIV infection by CCL5 utilizing isolates. There's a significant body of evidence that suggests a very positive influence of CC chemokines in the context of existing infection in adults. And there are also prototype studies in rhesus macaques that show that the production of CC chemokines by CD8 T cells or even other cells are associated with protection from infection. So there's some evidence to suggest that they're good to have. So what happened when we looked at the levels of production in infants? Okay, so here what we did was we isolated cord blood mononuclear cells, and we stimulated them with PHA, which is phytohemagglutinin. It's a lectin that non-specifically will stimulate cells, and basically looked at how much is produced. Here there are 4 groups here. The negative controls, these are infants born to HIV uninfected mothers. These are infants who are born to HIV-infected mothers but remained uninfected. So they're EUs or exposed uninfected infants. These are intrapartum infected infants and in utero infected infants. You can see compared to the controls, there's this big increase in the amount that is produced in infants who remain uninfected compared to controls. But the interesting thing that stood out was the intrapartum infected infants. They showed this deficiency in production. This was despite getting post-exposure prophylaxis of 1 dose of nevirapine in this particular cohort. The in utros, these children are already infected at that particular time, and that's a bit of a confounder because you're already, infants or adults who are already infected will churn out lots of different cytokines, so it's very difficult to assess exactly their ability to produce that. So the real comparison of these two, because at the time when we look at these in cord blood, these children are actually uninfected at this stage. They become infected through basically being during delivery. So the question we asked is, well, is there something that's different about the exposures between these two groups that would account for why these infants are producing less? So it's, in a way, they look like the controls. It's almost like they haven't had this ability to produce more. Or is there some inherent inability to produce that chemokine, which could be that it could be because of genetics, host genetics? So the first thing we did, we found no evidence really that there's any difference of exposure between your exposed uninfected and your intrapartum infected infants. And if you look here, what we did was we took cord blood plasmas from the infants and we tested them for various soluble activation markers, neopterin, beta-2 microglobulin, and soluble selectin. And that would really give an indication of the extent of activation in those infants. As you would expect, in utero infection, you'd have a lot of activation, and that would not be unexpected. But you have higher levels of activation compared to your negative controls, but these two groups are all the same. So it certainly would seem that it's not different exposures that would explain that. So then we thought, well, how do we ask the question? So we thought if inherently they are able to produce that, then their mothers may show signs of that too, and that's exactly what happened. You can see this diminished response again in the mothers that transmit intrapartum to their children, suggesting that there's some genetic mechanism that is underlying this. So then that made us say, well, okay, so what are the genes that code for this particular molecule? And we found ourselves on chromosome 17 in a very complex area of the human genome where you've got lots of different copies. And they've been segmental duplications over time. So there are 2 genes that code for the functional molecule. One's known as CCL3 and it occurs in 2 copies per diploid genome. So most genes you would have would occur at 2 copies per diploid genome. However, this molecule here, known as CCL3LA, or it's often known as CCL3L1, occurs in different copies in different individuals, different copies in different populations, and ranges from having no copies to individuals having, in fact, we've gone as high as seeing 13 copies in some individuals. There's also another gene, but was previously known as a pseudogene. It's not a true pseudogene. A pseudogene would imply that the gene is not expressed. This is expressed, but it produces only messenger RNA. It's not translated into a protein. So it's thought that there may be some function of this, but that is not known. The interesting thing about this particular molecule that occurs in different copies, it is the one that is the most potent natural molecule that is able to inhibit HIV replication. It's 30-fold more potent at inhibiting HIV compared to this molecule here, yet they are very, very similar. There's 96% amino acid similarity between those proteins. Not only is it the most potent at inhibiting HIV, it's also very potent in all its activities in terms of chemotaxis in its natural role. So just to show you how different these genes are in different populations, we've looked at Black South African individuals, and here you can see the number of copies range from 0 to 11 here. You can see the median is about 5 copies. So this is the frequency or the number of individuals that actually have these numbers of copies of that particular gene. If you look at Caucasian individuals, you see it's a whole different spectrum, and you see the median of copy number is 2, and there's a lot less of a breadth. What was very interesting around about the time when we were doing this work, and we just started looking at copy number as a factor in our results, a paper came out in Science showing that copy number lower than the population average, and you have to compare it to the population average, is associated with markedly enhanced HIV/AIDS susceptibility and disease progression. If we go by that alone, if we look at our populations and we say what proportion of individuals are below the median in our Black South Africans, you see that about 45% of individuals are there, whereas in the Caucasian population, you have 22%, about 23%. So the question, again, I think one has to always be cautious. We're talking about one particular factor. There are always many things that contribute to susceptibility. But if you had to base it on this particular molecule alone, one would say that there may be a greater susceptibility amongst our Black South African population compared to Caucasians. What we then did was we tested on using much larger sample numbers and basically showed that in infants who became infected, their copy numbers were significantly less than those infants who remained uninfected. What came out of doing the genetic studies was that we saw that it didn't make a difference whether they were intra or intrapartum infected. A lower copy number made infants susceptible through both those routes. We've also looked in the HIV-infected mothers, and we've seen that copy numbers also relate to higher copy numbers are associated with higher CD4 counts and lower viral loads, and also stronger T cell responses, both CD4 and CD8 T cell responses. So overall, looking as though these copy numbers do translate to importance in both protective immunity as well as attenuation from disease progression. So going back to our graph originally, we were really looking at the main comparison here, the exposed uninfected and the intrapartums. When we looked at the copy numbers and tried to see how that related to the production levels, we divided the exposed uninfected infants into those who had copy numbers, higher copy numbers and lower copy numbers. And you can see if you've got higher copy numbers within that group, you have higher production, which is what one would expect. But interestingly enough, in the intrapartum infected infants, even those that had copy numbers that could be detected as higher, they were completely unable to produce protein, which suggests that copy number on its own, yes, plays a role, but there's more to it than that. And you cannot take one copy does not equal another, and we've coined the term not all copies are created equal in the ability to produce protein. And we think, and we're trying to look at that still to prove that exactly, but we think that these infants may have more of the pseudogene, which is also picked up at the assay and allows you to quantitate the number of copies that there are. And they may just have more of these defective or non-producing copies. We've also tried to understand gene variants. So we've looked at both genes, the CCL3 and the CCL3, in South Africans, and have also published that work. It's very complicated when you're looking at variation and you've got different copies of genes in different individuals to try and get to the bottom of all of these things. So let's just look at what role might these particular molecules be playing. I think a lot of times what is often touted is really the role of these chemokines in their ability to block HIV infection. And there are a multitude of functions that could actually be involved in protection or in attenuation of disease progression. There are a number of antiviral mechanisms. This would be your typical non-cytolytic inhibition, blocking entry or down-regulating CCR5. There are also post-entry inhibition possibilities. And there's also the upregulation of various intrinsic antiviral factors. So these molecules have the ability to upregulate ApoBec3G, for example. One thing we thought quite interesting, but this hasn't been proven for HIV, and it's something we might want to look at, there's a direct antiviral activity where in herpes simplex virus, which is also a membrane-bound virus, any of these molecules, CCL3, 4, or 5, have the ability to generate pores, literally make pores in the membrane and kill the virus in that way, and that's rather interesting. The main functions of your chemokines are really immune modulatory mechanisms, and obviously in acute inflammation, chemotaxis is important, and it's very, very important. The abundance of a particular molecule at a site of infection can be crucial in terms of directing which cells come into the response, in what order they come into the response, how many come to the response, and that can really change the face of an immune response. So that may be a very, very important function of these molecules. Also, they have the ability to instruct adaptive immunity, and that's like an adjuvant effect. So if you have chemokines, these days there's a lot of interest because chemokines are very specific ways of driving the immune response. And you can almost tailor-make the response you want with a particular chemokine if you know the knock-on effects of that particular chemokine. In certain studies that have been done with herpes simplex virus in mice, they found that CCL3 promotes IgG, IgM responses, antigen-specific one, T helper cell responses, and various mucosal and systemic cytotoxic T cell responses. And all of these are the responses that everyone thinks are the responses that should be in a vaccine to HIV. So what we've proposed in terms of these molecules, and this is a very simplistic sort of model of that, is that differences in host genetic background, and particularly of these genes, would present the host with different abilities to drive HIV-specific immunity on primary exposure to HIV or even to HIV vaccines. So that individuals, and we've almost termed it, you have a natural adjuvant ability. So your ability to drive that is encoded in your genes. The important thing, I've just put CCL5 there, that we don't forget that these molecules really, their main functions are through CCL5. So it's this axis that may be very, very important in terms of this. So just an example here. For example, if you say somebody's got a particular genotype, so you've got certain copy numbers that gives you a certain amount of production of that particular protein. If you produce a normal amount, say wild type production, say your innate, this chemokine response is that, your adaptive immune response that. If it's someone who has a deficient production, as you see in the case of our intrapartum infected infants, for example, one might expect that if you were exposed and you had to develop an adaptive response, you might do that deficiently too. And the idea for a vaccine would be that this could act as an adjuvant and restore the situation where if you had a deficient innate response, it could drive. A very simplistic model, but and one that would need to be tested. And there is some evidence for certainly both these chemokines as well as CCL5 being very, very important in T cell activation and development of T cell responses. So there's evidence coming out to that effect. So I thought, again, just to highlight that is an important axis, and I think we all probably have heard of this. And this was the whole question of eradication of HIV in a case of the HIV-infected man who had acute myeloid leukemia and received 2 stem cell transplants, one February 2007 and then another one 13 months later, from the same HLA-matched but unrelated CCL5 delta 32 homozygous donor. So we know that this donor had no CCL5 expression at all on their cells. This individual remained uninfected in the absence of heart for 20 months. This was at the time of the New England Journal of Medicine paper. Viral rebound you would normally expect would have happened. So this was a lot of concern, a lot of case for excitement here. A further report, which has just come out now in April of this year in blood, showed still no evidence of HIV infection, and this is now more than 4 years later. So this is considered as the first proof of concept that HIV can possibly be cured, and although it's not practical in approach, has injected renewed interest into the possibility of HIV eradication. And I know there are a lot of questions around, even though some virus is undetectable, is it truly not there? And I think obviously this is going to have to stand the test of time to see how long there are no signs of infection and if that holds true. So I'm going to move on now to talk a bit about the NK responses that we've seen to HIV peptides. And this came as a completely unexpected finding. While we were studying HIV-specific T cell responses, CD4 and CD8 T cell responses, and trying to understand their role in maternal infant transmission, we were using a whole blood intracellular cytokine assay that measures interferon gamma and interleukin 2 in the cells. So if a cell gets stimulated, you would be detecting those cytokines. And the stimuli that we use are peptide pools, HIV peptide pools, GAG, POL, NEF, REG, and ONV. The regulatory region, we combine all of the regulatory regions into one pool. So this just gives you an example. Each of these pools is made up of a whole lot of overlapping peptides. They're about 15 amino acids long, and they overlap by about 10 amino acids. So there's about 66 in this pool, 92, 50 for each of those pools. So basically, when you give these pools to the cells, you're asking them, have you seen any of these peptides within these pools before? So is there some kind of memory? I mean, that's the normal process for this, a T cell response assay. So to our surprise, we saw these robust responses. In fact, we saw it in an exposed uninfected infant first, that we found were actually CD3 negative lymphocytes. CD3 is the marker that marks all T cells. So, and then later we determined them to be NK cells. We found particular markers. Once we started seeing this was consistently happening, we put in markers for NK cells. So you'll see me refer to them sometimes as CD3 negative lymphocytes because all the original data was just on those. They're CD56 DIM. CD56 is a marker of NK cells. The CD56 DIM subset of natural killer cells are cytotoxic cells. So they play an important role in killing virus-infected cells. And what did we find about these cells? What was really interesting, when we stratified and we looked at how many of the non-transmitting, 57% of the non-transmitting mothers had these responses. Only one transmitting mother had a response. And then also the exposed uninfected infants, significantly more infants had those responses compared to the one that ended up becoming infected there. And when we adjusted for maternal viral load, the responses in the mothers still remained significant. There were no other factors, not maternal CD4 count, not mode of delivery, not infant birth weight, or antiviral regimens that explained this association. And also these responses were not detected in any uninfected controls, not mothers or their infants. What was also interesting when we looked at the HIV-infected mothers, the mothers had lower viral loads and higher CD4 counts if they had a response. Also, if you had a response to, these responses were mainly to the envelope region and to the regulatory regions. And where mothers had both an ONV, or where there were both ONV and reg responses, your viral load is lower than having a response to either region alone, and CD4 counts higher. What was further interesting is that we could map these responses down to individual little peptides, so these 15 amino acid portions right across the entire HIV genome. We mapped 5 individuals' responses, and you can see, sorry, these are CD8 and CD4 T cell responses, NKs, and you see that the NK responses come up sometimes in regions where you've also got T cell responses. In others, you've had only those and you don't have T cell responses. A lot of variability in the specificity of the responses for different individuals, and this is really very characteristic of what you would find if you were mapping CD8 T cell responses. What was interesting was it gave us a look-see at which of the regulatory regions are actually the ones being targeted, and VPU came up and TAT came up. In fact, this particular TAT response is the only TAT response we've seen to date. In general, where we have regulatory responses, and they occur a lot less often than to the ONV region, they all are generally VPU. So VPU looks like quite an interesting target in terms of these responses. This one individual here, NP001, is a woman who was infected already for 10 years. And these are the amino acids. This region here is covered by about 7 overlapping amino acids. And her response is this response here in the TAT region as well. And we had the opportunity to get her back about 3.5 years after we did this first mapping exercise. So we thought, okay, let's have a look at them. So here, you can see these were the responses at the first time point. She had quite a broad response, a response to GAG, POL, NEF, REG, and ONV in terms of her CD8 cells, and 4 out of the 5 pools to the CD4 response, and this very robust response to the regulatory region and to ONV. At the second time point, what we did was we broke them down to just the region. So we did the full pools, and then also just the region, the 7 amino acid regions that actually responded. And we saw the same robust responses, and exactly the same region. So this is a very durable response. And this woman obviously has had an undetectable viral load, very good CD4. What we've subsequently done We've been working on a very small group of long-term non-progressors and elite controllers. This is just a group of 10. 9 out of 10 show a response of this nature, whereas when you compare it to the chronic infection sort of our mothers in this particular cohort, you'd expect on cross-sectional HIV-infected individuals, you'd probably see about 50% may show these responses. So you can see it's overrepresented in individuals who have very good control of HIV infection. So what could these responses be? Well, we know that there's CD56 DIM subset, and those are your cytotoxic ones. And there are two ways. You either have direct natural cytotoxicity, and this is where you have a natural killer cell will recognize, if you have a natural killer cell and a healthy cell, you will find that there will be no interaction there. Your natural killer cell, the minute it recognizes that your cell is infected or there's something or transformed, that will result in killing. I'll explain a little bit more about the detail of that later. The other way is an indirect system where an antibody forms a bridge between a natural killer cell and a target cell, and your antibody will specifically see HIV on the surface, be it on the surface, and will bind to FC receptors on the NK cell, and that interaction will lead to activation of the NK cell, which will then kill the target cell. So you either have direct or indirect mechanisms. So what do we know about these mechanisms to date? Well, we've done some work that shows that if we have NK cells from an individual and we purify them and we purify their autologous, their own CD4 T cells, but we give back their own plasma, they will actually, you'll see that gives you what is required. But you need a cell-cell interaction. It seems to involve class 1 molecules, HLA class 1 molecules, because if we add antibodies to HLA class 1, it actually inhibits that response. The interesting thing was there seemed to be a need for a non-specific soluble factor in plasma, which seemed to imply that there's a direct mechanism involved, that involves HLA, that involves something else in the plasma that's necessary. Because if you remove the plasma from the infected person that has a response, but you add back uninfected plasma, you can reconstitute the response. There's another group who also bumped into these responses, and they found that HIV-specific IgG can mediate it. So this is indirect, and this is antibody-dependent cellular cytotoxicity. Saying that, there certainly is, in our long-term non-progressor group, there seems to be an indication that certainly a number of them may be acting via this mechanism. But there are some mixes, so it almost looks as though there may be direct or indirect mechanisms. There may be a mix of those mechanisms. It's looking fairly complex. Okay, so this is a scary slide, but I just want to indicate just the principle of a question of balance. In order for an NK cell to function, it has to engage with another cell and recognize what they call ligands. So you've got a whole bunch of different receptors that you find on the NK cell. They'll engage with very specific receptors or have a way of binding to those. So you need those interactions to take place. Not all of them. And it's possible that you have interactions with some and not others. But under the normal non-inflammatory conditions, a natural killer cell is very strongly inhibited. And you have what they call inhibitory KIR or killer immunoglobulin receptors that interact with a self-peptide and an HLA. And as long as they see that, they recognize that that cell is not infected and they're not going to react. Under conditions of activation, when an NK cell will react, is when you have what they call missing self. So a lot of times infected cells will have downregulation of HLA, and this receptor will recognize that it's not there and say, this is the cell we need to kill, okay? A lot of times viruses do that to evade the cytotoxic CD8 T cell response, because you do need HLA on the surface for those responses to take place. But NKs, so it's almost a backup system of the immune system to say, well, okay, if you dare to downregulate that, you'll be zapped by an NK cell. The other thing is induced self, is where you have host proteins that are upregulated. So you have higher levels of particular receptors that are on the surface. A lot of cancer cells often have that. And your NK cells then first recognize that as a stress response and will kill the cell. And then obviously foreign recognition. So if you replace that self-peptide with a foreign peptide, so the peptides like, for example, that we're testing in the assays, if you put one of those in there, that would stop this inhibitory reaction and result in activation. So I'm going to be talking a little bit about the KIR and the HLA, because it's these combination of molecules that play a very important role in control of this interaction. But they've also been ones that have been shown to play a very important role in control of HIV infection in the literature. So of course, just to add insult to injury, we find that these molecules if we look at the genes, find themselves in the two most polymorphic, the two most variable regions of the human genome. And you can see here you've got your class 2, class 3, and class 1 molecules, HLA, A, B, and C molecules that are all coded for on this particular chromosome and on a completely separate chromosome. And you find that over evolutionary time, these have been evolving separately. And the fact that there is such variation, and in African populations, there is greater variation than in various other areas. It shows that there have been these evolutionary pressures on humans over evolutionary time. And these are the things that also code for the different susceptibilities in populations. So just to show the diversity we have in our populations, this is our whole mother-child cohort where in the end we've tested about 440, we've genotyped about 446 samples of mothers and infants. And these show the KER genes, so your killer immunoglobulin receptor genes. We've got 14 genes and 2 pseudogenes. So you have inhibitory genes, which are these 2DL1, 2, 3, 4, and 5. Activating genes, S1, 2, 3, 4, and 5. You've got pseudogenes that exist, and then you've got various framework genes that exist in all your KER genotypes. Some of these are important in terms of how they bind. So various HLA molecules, class 1, your HLA-C molecules, that are all divided into either C1 or C2 molecules, and they very specifically bind to some of these particular ones. I'm not going to go into, obviously, any details on those. And then one that's very important is also the amino acids that define certain HLA-BW4 types. These have shown to be very important in relation to this particular one of these genes in terms of control of HIV infection. So this little scheme here shows this. These are all the different, and these are just patterns of possession of genes. So going from 7 genes to as high as 16 different genes and all different combinations. And these are 46 different KER genotypes just in our own population to date. The ones that are in blue, I've just marked them because they're about more than 5%. The prevalence is about more than 5% in these particular cohorts. So these are the ones that are much more prevalent. This particular one here, which is one of the lowest numbers of genes, the AA1 haplotype, is the most prevalent haplotype in our populations as well as worldwide, occurring in about 26% of individuals. So that's a very high prevalence occurring genotype, and I'm highlighting that because I'll touch on that shortly. When we looked at our HIV-infected mothers from this cohort, we actually found those that had responses to HIV peptides had quite different profiles when we looked at KER and HLA. Okay, and here this just really shows the KER genes. And what we saw was individuals who have responses tend to have significantly higher numbers of these genes. So anyone with 13 and above genes, 64% were responders versus 32% of your non-responders had those. When you have higher numbers of KER genes, you have higher numbers of activating KER genes within those genes. And it's generally held that individuals who have a higher activating genes are more able to control infectious diseases. However, what is often problematic is you are more likely to suffer from autoimmune disorders. And that's really because your NK cells are just more highly reactive. So you may be more able to fight infectious diseases, but more prone to autoimmune. What was also interesting, there was a particular genotype which was extremely unusual, which really stood out. And that was this one here, which has 3 activating genes. And that obviously just highlights an area that we can look at more closely to see what it is about this genotype that will allow individuals that have this 13 gene genotype to actually respond to peptides. So these are all giving us clues about what the underlying mechanisms are of these responses. So one thing that we did was we took the group, the AA haplotype, the one that has those few numbers, the one that I highlighted that's in about 26% of the population. And we thought, well, okay, if we look at individuals who've all got that same genotype, because it is fairly prevalent, and we managed to get 43 individuals together that have that, and thought, well, let's look more closely at the HLA that bind and that are known to bind to some of the KERs and see if there's any role there. And the other thing we could do is it has only one opportunity for an activating gene there, and that's this 2DS4. So we thought, well, that will allow us to look at the variation actually within that gene. Because it's not just that you have a gene, it's also the variation within that gene. So when we compared responders and non-responders, individuals, there was no activity on the HLA-B whereas all the attention has been in all the studies that have been, it's showing in our populations that HLA-C is really important. And here you can see individuals who've got responses of much, much more of the C1 versus the C2. Okay, I'm going to, yeah. And then, and here you can see, so basically showing that particular alleles that fall into these categories play quite a significant role in the ability to respond. The other thing that came up was just that if you looked at the activating gene, there are some that code for full-length protein and some for a truncated protein. A full-length protein ends up being membrane-bound, whereas the truncated one is a soluble form. And that then also seemed to show this shift in that responders tended to have more copies of the one that was on the membrane, showing possibly a greater opportunity for activation and therefore a greater likelihood of immune responses in that group. Almost done. Then when we looked at mothers and infants, we went back to the mother-child cohort where we looked at the responses that we know are associated with protection. We had a group of mothers where both mother and child had responses, but there was also a group of 22 where the mother had a response but the child didn't. And because of the genetic relatedness between them, we thought, well, why do these infants lack responsiveness? Can we pick up any clues from comparing the mother and the child? What was interesting, gene content here was completely comparable. Mothers and infants were similar. Here, there were reduced numbers of genes. So it was completely in agreement with previous thing, and more haplotypes that had much lower numbers of activating genes. And also, the protein, which is not, which I said is a truncated protein or a soluble form, they had more of that soluble form compared to the membrane-bound form. So they had the same characteristics, and these are all infants that aren't infected, as the HIV-infected women, where we looked previously, and so certainly showing that KIR2DS4, the one activating receptor, matters, and the particular HLA class 1C molecule. All right, I'm going to skip that. That's just going back to the model and what we think might be happening, because just overall, I'm just going to conclude by saying we think that there's engagement of multiple receptors and an integration of a whole lot of different signals, that it's not just going to be one mechanism that explains how these cells can respond, because there's so many opportunities for them to be activated through different receptors and different combinations. So our task is really to try and continue to unravel that. Right, so this is just to conclude. In terms of going forward, mother-to-child transmission, because of the increased use of antiretrovirals, we're no longer able to go out and get new cohorts now to study protective immunity to HIV. I mean, that's increasingly becoming difficult. However, because of the material that we've collected over many years in different cohorts, we will continue to do genetic studies on stored material. In terms of long-term non-progressor and elite controller studies, the importance here is understanding good control in existing infection will help us to identify mechanisms and pathways that can be targeted for new therapeutics. This is a very interesting area, and if anyone has interesting patients that would fit into those categories, please contact us. And then in terms of healthy control studies, we're doing a lot of work where we're trying to understand how host genes might impact on immune responsiveness. And here you want to do it in the absence of a chronic infection, which obviously then also modulates immune responses. So to see, for example, that axis between the CCL3 as well as CCR5, I mean, does that indeed show differences in people's ability to respond to various vaccines? The whole question of NK cell memory, probably very difficult to finally prove in humans. In the mouse studies, it was the natural killer cells that reside in the liver that were the ones that actually had the memory response. So it's quite a challenge to try and determine what's going on in humans. If there's memory in NK cells, what about other innate immune systems? And how can innate immunity be manipulated to provide protection or also to improve therapeutics? We also have an aspect of mucosal immunology. We do various studies on lactobacilli, their interactions with the immune system, and we're also looking at various live microbicide approaches to treating sexually transmitted infections. So that's just another aspect to some of the lab work that we do. And then just to acknowledge my team of researchers and various PhD students, MSc students, and collaborators over the years, and clinical collaborators working on all the mother-child cohorts. And then, of course, thank you to the funders, from the Wellcome Trust to the NIH to all the local funders. Many thanks for your attention. Thanks.